Dr. Evan Nadler from Adipomics. And so, Dr. Nadler, if you want to share your screen and tell everyone about Adipomics.
Thanks, Jennifer. So thank you for the opportunity. My name is Evan Nadler, and I'm a pediatric surgeon scientist at Children's National Hospital here in Washington, D.C., where me and my co-founder and research partner, Rob Freistadt, started Adipomics, which aims to unlock the potential of the adipocyte. With respect to this particular pitch, the fundamental issue is that third trimester gestational diabetes testing is too late. Why is that so? Well, we do testing at 24 to 28 weeks for gestational diabetes, mostly because we can. The oral glucose tolerance test hasn't been changed in over 70 years. And if anyone on the meeting has actually taken that test, you know, you drink some awful liquid, you sit there in the office for a couple hours with an IV in your arm, and you get a couple blood tests. And the whole experience is not particularly great. Not only that, it doesn't actually address the real problem or the entire problem of gestational diabetes, which is the intergenerational or transgenerational passage of obesity and diabetes to the fetus. So sorting that out, making it simpler. In a pregnant mother, we're doing a test in the third trimester to make a diagnosis of gestational diabetes, where the injury to the fetus is actually in the first trimester. So we're completely missing the boat. We all know that gestational diabetes leads to maternal diabetes and big babies, but it also leads to childhood obesity and type 2 diabetes in childhood and adolescence. So the current paradigm is unworkable. So what's our solution, or why are we so smart? Because I'm sitting in a closet. That's why I'm so smart. What we've determined back in 2015 through our research efforts, and I should say that Rob and I were just your basic, ordinary, basic scientists working in a collaboration trying to understand disease. And what we found is that fat cells or adipose tissue can excrete these small extracellular vesicles, also known as exosomes, that carry genetic information through the circulation to all the distant organs throughout the body. And in these exosomes, or these transport vehicles, the microRNAs actually turn on the pathways in the end organs that cause disease. We started with isolating these fragments from fat tissue, but now we can do it in both blood and urine. And actually, urine turns out to be the greatest yield of adipocyte-derived exosomes, even better than blood. So we aim to replace the current OGTT, an awful test with multiple blood draws, with a urine-based test, which will be a reference lab-based assay to detect a disease predicting microRNA signatures, which in the end will lead to earlier intervention using Alonzo Wellness, hopefully. This is true for any disease, by the way. We're talking about gestational diabetes today, but we actually have identified a core panel of microRNAs that just show that your fat is misbehaving or sick. And it turns out that around this core, there are also secondary microRNAs that sort of dictate whether you get fatty liver disease from your sick fat, or diabetes from your sick fat, or actually even atherosclerosis from your sick fat. We can talk about that later. So in terms of the gestational diabetes panel, we've identified about 18 microRNAs from adipocyte-derived exosomes found in the maternal blood of mothers, pregnant moms, at 28 weeks gestation. And these microRNAs are not only upregulated in patients with gestational diabetes, but also drive abnormal cardiometabolic development in the fetus. So again, sort of to try to boil it down, nuts and bolts, these are the microRNAs that turn on the pathways that cause these diseases. So as I mentioned before, the adipocyte-derived exosomes are actually easier to extract and found in greater numbers in urine. and most patients don't mind giving multiple urine samples. So we envision a series of tests over the lifespan of the pregnancy where we can monitor progression of the intervention. And we're working on our 8-week data currently, as our first panel was created based on the 28-week data. And the science here is patent protected, with the patent you see listed and a couple other that are also pending, not shown here. So this is just one example of the kind of work that we're doing. And what this is a Keg graph or a Keg diagram. And if you pay specific attention to mere 4649 3p, you don't have to remember that one. It's not, it's not important other than to say, to illustrate this point. May or may not be part of our secret sauce, but I'm not telling you. What you see with those big red dots is that the red shows that this particular microRNA is statistically significantly different than in patients without gestational diabetes. And it impacts the signaling pathways that the arrow points to, which are all stem cell related pathways. So the implications of what's going on here are far greater actually than gestational diabetes. But this is just an example. So our timeline. So as I briefly mentioned before, we started as basic scientists working in a lab. And it was when we found that we could isolate these adipocyte-specific derived exosomes from urine that we realized that there was a company in the making. Our first success was winning the NSET 2 University Startup Demo Day back in 2016. And then we actually became a JLabs company in 2017 once we really understood the importance of our findings. 2019, we got a grant from the NIH and the Fogarty International Center that actually is the basis for all these data, which is, you see the name of it there. And if anybody wants to discuss the thin fat Indian baby paradox, we can do that in the session afterwards. It's a very complicated clinical scenario where the babies are born with low birth weight but high adiposity. And then, I think we all recognize that particular logo of the SARS-CoV-2 virus. So everything stopped in 2019. We weren't allowed to do any research at our hospital that wasn't COVID related. So we actually pivoted and formed a JV to create a SARS-CoV-2 diagnostic that actually is about to undergo its our testing for our FDA submission, which we hope to exit soon, and then use those funds to funnel back into our core business, which is Adipomics. We moved to the new JLab site here in DC, up at the old Walter Reed Medical Center. And in 2022 and moving forward, not only are we going to confirm our gene signature and roll out our diagnostic, hopefully in the next 2 years, but we're also working on therapeutic options where we use the power of the exosome to overcome some of the known difficulties of lipid nanoparticle administration of drugs and genes. Again, we can talk about that later. This is our management team. Rob is the brains. I'm the looks. We got Keith and Tom, who are our business strategists and helping us bring it to market. And then our mentor, Eric Hoffman, who is president and CEO of his own company, Reverigen, which is doing its own good business with steroids or steroid-related medications. So with that, I thank you for your time, and I'm happy to take any questions.
I was wondering, can you help me connect the dots with kind of the immediate implications for clinical care once you have this fully developed? Like, how do you see this used?
Sure. So going back to the sort of initial paradigm, by the time you make the diagnosis of gestational diabetes, the cat's already out of the bag. Your fetus is already genetically imprinted, and the risk of childhood obesity and type 2 diabetes in childhood or adolescence is already well past. So our aim is to make the diagnosis, or at least predict the diagnosis, much earlier during organ development, So in the 8-week range. And then you can intervene with, be it lifestyle, be it medications, be it apps, whatever you choose to intervene in with. And then you can monitor with a serial urine-based test, let's say monthly, to see if your expression of the microRNAs is moving in the right direction or the wrong direction, and then pivot accordingly. So basically, we're just trying to, and this works actually for all the diagnoses we're tackling. The aim is for earlier intervention. The aim is to detect plaque disease in your arteries before your cholesterol is 400, not after you get the blood test and make that diagnosis. We want to tell you when you're depositing plaque long before your blood tests are abnormal. And the cases here in gestational diabetes, we're going to tell you that you are trending towards gestational diabetes, 20 weeks earlier than the standard of care, the current standard of care.
I see. So maybe you start metformin, maybe you have an exercise program.
We're not going to certainly dictate the intervention. That'll be up to the practitioners, but at least we're giving the information to both the patient, the pregnant mom, and the practitioner much earlier than they would normally get it.
So kind of a risk profile, like you're not diagnosing gestational diabetes, but you're saying this person is high, medium, low risk for gestational.
We're diagnosing sick fat, and that sick fat is bad, is a sort of, is known, sort of. So we are diagnosing microRNA changes in your fat. So there is a diagnosis, but those microRNA changes predict disease, predict overt disease later on.
Let's see. We have a question about whether you think this technology could similarly be used to predict risk of preeclampsia. I don't know if that's an application you've thought of.
The answer is probably yes, that it could, although, I'm a pediatric surgeon and Rob is a ER doc by medical training, and we're both NIH researchers by night. So neither one of us, can I say, is an expert in preeclampsia, but it certainly can predict hypertension, and it certainly can predict cardiovascular disease. So I would think it probably could do the same. If there's anybody out there who has a big cohort of patients that wants to give us some samples, we're happy to look into it.
That's a related question, is whether you have an OBGYN on your advisory board or some other connection, because those seem like those immediate implications where the outcome comes in, at least, some portion of the outcome can come relatively soon with a birth. So
sure. So the short answer is no. The long answer is that Rob and I started this journey looking at, type 2 diabetes, just regular old adult type 2 diabetes. And it wasn't until our NIH grant with partners in India where we started looking at gestational diabetes. So it's somewhat of and like I said, we have we've shown changes in the respiratory system, the liver, diabetes, and the heart. So we need to build out our advisory board, and certainly be happy to discuss that with folks offline. But again, the paradigm is sort of earlier. The paradigm is personalized medicine, because we're looking at your particular adipocyte exosomes and disease prediction. How is your sick fat going to affect you? And then what can we do earlier to try to prevent that from happening?
Dr. Eisenberg, we've got time for one more question. So if you want to pick from the group.
Sure. Let's see. So I guess kind of a more technical question is just how are you detecting these exosomes? Like, is this a PCR type diagnostic? And apologies for a lack of technical understanding.
Sure. No, so so there are lots of exosome isolation companies, and I think there was one on Pitch Club a couple weeks ago. And so exosomes are hot now, and people are all over this field. And so what we do differently is, one is we specifically select out the adipocyte exosomes. So we're selecting signal out of the noise. And then the characterization is just gene chip, regular old microRNA detection. Eventually, we'll put this on a card, and we're having discussions with a couple folks who can do this, where it'll be like a colorimetric assay where the card changes color. The 8 genes or 10 genes or whatever the number is change color, and then you get if 5 out of 8 change color, it means your risk is 70%. If 8 out of 8 change color, your risk is 100%. And if 0 out of 8 change color, your risk is 0. Something like that. There's work to be done, but it's mostly in the interpretation and the algorithms to analyze the data, as opposed to the technical aspects of isolating the cell fragments and characterizing their content. That's fairly standard off-the-shelf stuff.
Okay, thank you.
My pleasure. Thanks, everybody.